Hello, welcome to demola. In today's lesson, we are covering protein structure. Very important, we're going to go from primary structure all the way up to quaternary structure, linking back to ideas of protein synthesis as well. No time to waste, let's get straight into it. Okay, so if we're going to talk about proteins, we need to talk about the building blocks. What actually makes up a protein? And that will be our amino acid. So I'm opening it up. Cool. So amino acids are a monomer of proteins, yeah? And you have your essential amino acids and your non-essential amino acids. So essential, not made by the human body. You need to get these in your diet. Non-essential, they are made by the human bodies. That's fine. You don't need to know specific examples for either or, not really relevant to your A levels. Cool. All of your amino acids have this general structure where you have an amine group, an R group, and a carboxyl group all around a central carbon atom. You need to be really confident with being able to draw this diagram. If they ask you to draw a specific amino acid, more times they will give you what the R group is, and you just have to add that. So like, for example, say if we have amino acid and they tell me that the R group is just like CH3, for example, then I'm literally just drawing out this, and then that would just be okay for your exam. So literally the same thing, but you just replace whatever the R group actually is. Cool. It's known as variable. They call it a variable group. Cool. Now, R groups can be different. They make the amino acids unique. So all of them have the amine, all of them have the carboxyl, but they have different R groups, right? So your R groups can be hydrophilic, they can be hydrophobic, polar or non-polar, charged or uncharged. It's just going to be depending on what they give you. Yep, cool. What you also need to know is that to join your amino acids, we have a peptide bond. If you are doing A-level chemistry, you will also know this as an amide group. That is the year 13 topic. But you can see here the carboxyl group interacts with the amine group, forming this bond that we have highlighted in purple. And that's what we call a dipeptide. So two amino acids joined together. As you could imagine, tripeptide, 3 amino acids joined together, polypeptide, multiple. And and that's literally done now, and are forming in these condensation reactions, which is also very, very nice to know. And that kind of wraps up what we need to know. Okay, and when we talk about proteins, right, what we'll describe a protein as is a chain of amino acids. Yeah, cool. So, protein structure, there are 4 main structures. We've got the primary structure, as we've drawn now. Definition, important for exams, a sequence of amino acids joined by peptide bonds. And you can literally see that I've drawn out amino acids joined by peptide bonds. Literally, we could highlight them as well. Not going to take too long, but you can see that's a peptide bond. This is another peptide bond. This is another one. You see, it just keeps going on and on and on until you finish off that polypeptide. Cool. Your secondary structure means that we've got hydrogen bonds forming between different parts of the chain. So it folds into an alpha helix or beta pleated sheet. Please just remember those two. You can see them in the diagram. So you can see that hydrogen bonds will form between, normally it's between oxygen and hydrogen. So those groups exist. You can kind of see them in the diagram here between a hydrogen and oxygen. Yeah. And and also they can form between literally your hydrogen bonds is forming because of difference in electronegativity, right? But as you can see, different parts of the chain are forming hydrogen bonds, alpha helices, beta pleated sheets form. Your tertiary structure is where things get a little bit interesting. You're going to have bonding between R groups. This is another one that's important to know. So the bonding between R groups causes it to fold into a 3D structure. And you're going to want to say 3D structure, and you're going to want to say bonding between R groups if they ask you in an exam. There are so many different types of bonds we can have. Ionic, disulfide, hydrogen, London forces, hydrophobic, hydrophilic interactions. Just be aware of all of those are happening between specifically R groups, which is going to cause it to form a 3D structure. And finally, we've got the quaternary structure. That's the final 3D structure. So we've already formed the tertiary structure. We have bonding between R groups. We've got a 3D shape. And now literally, we're joining different tertiary structures together, or we're joining different proteins together so that we can have a fully fledged 3D structure. And you can see in the diagram, this is actually hemoglobin. So we have alpha chains joined to beta chains, both different protein chains that are being put together to form a quaternary structure. Before we call it a day with proteins, thought it was just going to be good to mention about fibers versus globular, a bit of a debate. Now, globular proteins, these tend to be spherical, compact, yeah? What you'll notice, the cause of this, although I've never really been able to understand it, so you probably don't really need to, but why they're spherical and compact is just because the hydrophilic groups are all on the outside, whereas the hydrophobic groups are on the inside, yeah? So what do we also take by that? If my hydrophilic groups are on the outside, yeah, this is going to mean it's soluble because I can then form these hydrogen bonds between those hydrophilic groups and water. Yeah, so water can then surround your molecule. Now, globular proteins, examples, enzymes, transport proteins. So, for example, our boy hemoglobin, which we've got on the diagram now, and all of your hormones tend to be globular. And the reason why these hormones specifically would be globular, especially like hormones and transport proteins, is due to that solubility. So much water in the blood plasma. Yeah, top of my head, about 90%. So that means that these substances can be soluble in the blood and they can flow with the blood to get to cells. And specifically, hemoglobin is really important to know. We've mentioned this before. We've got beta chains, alpha chains. Those are different polypeptide chains, 2 of each. We've also got each of those groups has a heme, which has a central Fe2+. And that's how it's going to be able to carry your oxygen molecules. Cool. Then we just need to talk about fibrous proteins. These are long-chained molecules. So it's just a repeated sequence of amino acids, yeah, making a long chain. And the hydrophobic groups are this time the ones that face outwards. So if it's hydrophobic, can't form your hydrogen bonds with water, so they are insoluble. And these proteins tend to be structural. So like you got keratin in your hair, you got collagen in your skin, yeah? Collagen is the one that's kind of important to know. Not in too much detail, especially not right now in the course. But you got your collagen molecule where you've got those amino acid chains kind of coiled around each other. And multiple collagen molecules will then form a collagen fiber, which is what makes it really strong and can act like, for example, a barrier in the skin. So that is what I would say know about fibrous versus globular. If I stuck to timings, this should be a nice, short and sweet video. We've got 2 questions for you to try, 2 4 markers. Have a read, have a go. We'll come back with the answer. Okay, so here are your answers. We're going to break it down. First of all, what are they actually asking me in this question? They said explain how a change in the primary structure of factor 8 could cause difficulties with blood clotting. Factor 8, what does factor 8 do? It tells me that factor 8 is an enzyme that's involved in the process of blood clotting. That's important context for me to know. So when we get to your answer, they're telling you that it's a mutation, right? There's a change in the sequence of amino acids. That's the first thing you want to say. There's a different sequence of amino acids. Now, if there's a different sequence of amino acids, it means that the position of the R groups changes. Yep. So that means that the bonding between the R groups changes. Or you can say there's a different tertiary structure, whatever you want. Yeah. Then we get the context from the question. I was told that factor is an enzyme. If we change the shape of the active site, then it can no longer bind to substrate, which means that we don't literally just anything. It even says here, I'll highlight in green, just mention anything about the blood clotting cascade. Use the knowledge that you already have to just add it in. A little sprinkle there, press the examiner, you can get a mark from it. The next question literally was just a comparison question between fibrous and globular. So what do we know? What's similar? We'll do that in green. Well, both of them are proteins, right? So amino acids join with peptide bonds. Happy. What else? We know that both of them have a tertiary structure, right? So they both have a tertiary structure. They then both must have these bonds that are joining the R groups, the hydrogen disulfide bridges, ionic bonds, etc. Cool. We mentioned this about differences, which we're doing red. Globular proteins, hydrophilic. Oh dear. In a globular protein, we said that hydrophilic is on the outside, and in fibrous, we said that hydrophobic was on the outside. That's the difference. Also, something that I didn't mention, probably should have, but you could have picked up 3 marks anyway. I haven't said that globular has tertiary quaternary structure. Fibrous have little or no tertiary structure. That's just something that's good to add on. Right? And then you could have got 4 marks on this question, yeah. This is another one. Globular proteins are folded into compact. Fibrous is long chain. Just keep to what we learned, right? Keep to what you've learned, and then just try to apply that into the question where and ask you. Okay, so a lot of our ASR tips today are going to be more involved in like different topics, if that makes sense. So like, for example, for this protein structure, you really, really need to understand protein synthesis. Like, it's so important to understand how proteins are made, because that is where you really are going to pick marks up more than just about describing different protein structures, if that makes sense. Also about join the structure of amino acid. If it comes up, it's easy marks. Different structures of protein as well. you need to be confident with those definitions, being able to say which is primary structure, which is tertiary, which is secondary, whatever. And then knowing the difference between fibrous and globular proteins and having examples for both. I would say, really, to be honest, if you get collagen and hemoglobin down, you're probably chilling. What do we not want to do? The steps of protein synthesis, so like your transcription translation, just don't get those confused, because it is an easy thing to do if you haven't covered it in a while. Also confusing these different protein structures. So like, for example, I'll say about secondary, you start describing primary. Just make sure that you know about that progression that the protein goes through to get to that from the primary to the quaternary, if it has a quaternary. Then also the structures of hemoglobin and collagen. It's really good to just be able to describe them. I think it gets overlooked a bit, especially with collagen. And then not being able to draw an amino acid. I've seen it happen before. Just make sure that you're really confident with drawing those. So that is going to be a wrap for the lesson today. Thank you so much for listening in. I hope it's really helped. If it has, please drop a like and subscribe to the channel. In the comments below, any questions I can answer. I will do my best to elaborate on it. We've got the TikTok and the Instagram both at demola. But that is all for now. I'll catch you in the next one.